And our next speaker is Luca Gerosa, who is a postdoctoral fellow in Peter Sorger's lab, where he studies signaling dynamics of ERK, of the ERK pathway in BRAF-resistant cancers. And I believe he is going to tell us something about how these dynamics contribute to cancer resistance, to drug resistance of cancers.
Okay, thank you, Nikolai, for the introduction and the organizers for this great conference. I need to apologize already. There's not going to be any single-cell proteomics data in this talk, but what I'm going to speak is single-cell data and hopefully a mechanism used by cancer that really relies on single-cell behaviors and therefore could be a good biological process that will be possible to study with single-cell proteomics. Maybe just to put you in the context of the cancer problem that I'm studying during my postdoc, which is adaptive resistance to targeted therapy in cancer, I just want to remind you that human cells transduce mutagenic signals coming from the environment through these cascades of signaling pathways that are made of membranes, receptors, adapters, and then, for example, kinases as a way to transduce proliferation and survival. And what we really know is a lot of these proteins get mutated in cancer. So they are a hotspot for oncogenic mutations, which can drive the growth of cells without the need of the signaling above. So the fact that these mutations drive cancer has been the reason for developing targeted therapies. So these are small molecules that can inhibit the oncogenic protein through, for example, small molecules or antibodies, and therefore block the growth of cancer. But you can see here already a problem. If a molecule inhibits the oncogenic mutations, the cancer is still the physiological pathway that can be used to compensate for the drug. And that's the type of problem that I'm going to show you in this talk. We're thinking, what is the role of these? And so the problem is understanding resistance to targeted drugs by adaptive reactivation of this pathway. And the particular cancers that I'm going to show you today are cancers with this BRAF V600E mutation. And here, graphically, I'm showing the status of a cancer cell that gets this mutation. So what you can see here is that this BRAF mutated component is able to phosphorylate the MEK and ERK pathway and drive proliferation of cells independently of what's happening at the receptor levels. In particular, the overexpression of this ERK kinase phosphorylation induces some negative feedback that blocks receptor signaling. So completely cell-autonomous process that transduce growth. RAF inhibitors have been developed for these cancers but are able to inhibit this mutation. And what happens is we have a rewiring of the network in which ERK signalings drop in time, and also the negative feedback, however, gets removed from the receptors. And here we have a case in which another pathway, which is not targeted by the drug, the physiological pathway of signal transduction, can take over. And the question really is, cancer cells that are treated by these drugs, how much can they reactivate this pathway? And how much is going to be dictated by how much growth factors there is in the medium and how much receptors these cells have. So we know that some colorectal and thyroid BRAF mutant cancers can actually reactivate this pathway dramatically. And these drugs don't work in patients that have that particular lineage of the BRAF mutants. However, melanoma patients respond initially for roughly 8 to 9 months, and then the disease reoccurs. And in my project, what I wanted to know, here I'm showing you data on cell lines. So we take BRAF V600E melanoma cell lines, and these are data from previous publications, add a RAF inhibitor, which brings down the ERK signaling pathway. And what you can see here is that the rebound we were expecting if this adaptive mechanism exists, it's very small. So 5% of the signal in the whole populations. But we had good reasons to believe that something was going on here that is not well captured by the whole population assay. So the goal was to understand what's happening really here in terms of adaptive ERK reactivation. And what we did is just to reproduce exactly that data, but using immunofluorescence. So now I'm showing you here, if you add one RAF inhibitors, vemurafenib is the name for it, on cells, melanoma cells, you have a drop in the ERK signaling, so the oncogenic-driven pathway, and then there is this tiny bit of reactivation when we look at the average. When we look at what really is there at the single cell level, however, now I'm showing you the distribution of single cells quantified by immunofluorescence. And you can see that already after 1 hour of adding the drug, there is this, every dot now is a single cell, and there is the appearance of these cells that are as high as the untreated control. So somehow this cancer cell reactivated the pathway. And when you look at how it looks in the immunofluorescence, this is red, this is the DMSO control, so no drugs. You can see that every single one of these cancer cells is driving the oncogene ERK very strongly. And now suddenly we have these colonies, kind of what they look like colonies or patches of cells in the drug treated that are as high as the other. So what's going on here? First of all, we want to check that there's not like a genetic mutant, with small clonality there, and it's not there. Sorry, I have to first say one other thing. We did a dose response curve here and found out that these single cells happen exactly at the type of concentration you can have in patients. So it's a relevant dose of the drug. And also when we extracted single clones from these cells, remade a population and reassessed the phenotype, now I'm showing you one of them, you can see that is perfectly matching. So there's not a genetic mutation. You can recapitulate both the high and the low states by starting from a single cell. When we used another drug on top of it to lower the maximum activity that these single cells can have, and then we measured the ability of these cells to enter the cell cycle, we figured it out that these high cells, what they do, they allow some cells to escape the G1S block that this drug do. So it's not important to match the details here, but these high cells seem to allow a small fraction of cancer cells to continue to proliferate. So potentially very important for the clinical side of it. We also used multiple cell lines. So here are 8 different cell lines and looked over 5 days. And what you can see that in all of them, untreated cells are very high phospho-ERK. And during treatment, you have these small amount of cells that start to appear. And then when it's quantified, is around 1 in 1,000, 1 in 100 cells in the population. So is this happening, for example, in vivo? What we did is to take the same cell lines and have xenografts, so put into a mouse, and then looked as a control or treated with this targeted therapy. I'm not sure how much you can see it here, but this is the control in which the ERK staining, so this is a whole tumor, and every dot is a single cell in millions of cells here. It's high in ERK before adding the drug, and then in the treated, what we found is again these in vivo, these small colonies of cells that are high. So all of this made us think that there is an adaptive mechanism that is working here that could be actually temporal. So there is something that turns on and off these cells. That was our hypothesis. And so what we did is to transform these cells with a reporter of ERK to do live cell imaging. And this reporter, developed by some other group, it's nuclear when ERK is low, and then it becomes cytosolic. It's pushed out from the cell when ERK is activated. So what you're seeing here is cells that have been treated for 24 hours with these RAF inhibitors, and so are properly inhibited. The oncogene is down. And now I just play this movie, hopefully. No. Here we go. And I want you to look here. What you will see is the emergence of spontaneous pulses in the population. So these are cells that are properly inhibited and then suddenly reactivate only for a brief amount of time their cancerogenic pathway. And when you quantify these on a larger field, and here I'm segmenting the cells and then plotting the ERK activity as color, you can see that there are pulses that appear in the whole population at different spots at different times. And for example, here there's going to be a big one. And what you basically discover is that the single cells that we were finding as patches are actually dynamics. When we look across the whole time and overlap the pulses, what you can see is that these are very high pulses but have a duration of only half an hour. And if we look for 15 hours, almost 1 in 4 cells is actually able to do one pulse or multiple pulses. So we had good reason to think that this is a paracrine autocrine signaling, but in which basically some cells can elicit the turning on of the cells near to them. And so what we wanted to do is to basically be able to reconstitute these pulses in the whole population by adding growth factors. And that's what I'm showing you here. So if you titrate the drug, the vemurafenib, for 24 hours and then add different growth factors, all of them can turn on the pathway in all the cells. And then if you look in time how this happens, this is exactly the same type of profile that we find in the single cell. So in order to understand if this was really due to this removal of the negative feedback that I told you before, what we did is proteomics. So this is absolute proteomics done with Steen Wiley at PNNL. And what I'm showing you here is the use of 5 different concentrations of the drug for 24 hours to tune the ERK pathway activity. And then for every protein in this pathway, I can show if it stays the same, as it happens for many of them, or if it goes down with the treatment. And the proteins that go down are in fact the proteins that allow now for this adaptive signaling from receptors to be transduced. So this was consistent with our hypothesis. And what we did is to turn our signaling data and this proteomics data into a kinetic modeling of the pathway in which there is the oncogene branch, there is the negative feedback that gets removed, and there is the ability of growth factors to reactivate it. And here I can show you this model. Once it is trained on data, it actually recapitulates and is able to explain what's going on. So when the oncogene is treated with increasing concentration of a drug, this is the black line. It brings down MEK phosphorylation and ERK phosphorylation, and cells are inhibited. However, when growth factors are titrated on top of it, you can see that only at the highest doses of the drug, we have the receptors being able to transduce the signal all down this cascade and reactivate the pathway very potently. So we use the model to ask, what is it that makes these cells only have a pulse, so only a transient activation? And when we ran simulation to find it out, the answer that came out was receptor level. So this red line here is the simulation of normal cells. And if we overexpress 10 times in the model receptors, what happens is instead of coming up and coming down as a signal, upon addition of growth factors, we would have a sustained signal. And if there are no receptors, of course, there's going to be absolutely nothing. And we tried these by using CRISPR-I and CRISPR-A systems to overexpress EGFR receptors in these cells. And this is what came out from the experiment. So these are 2 guides in the CRISPR-A systems that turn on around 10 times the EGFR receptors. And when we add growth factors, indeed what we have is a stabilization of the signal. And the model was predicting that this occurs because if you make enough receptors, you're going to have enough receptors at the membrane, even if there is internalization upon activation. And this is what we saw by looking at imaging data here. So these are the normal cells. You add EGF, and everything gets internalized and degraded pretty quickly. But if you have overexpression of EGFR, cells are, of course, have much more EGFR starting with, but they can continuously have EGFR at the membrane. So this explains why there is only a pulse and not a sustained signal in these cells. But we wondered, why is it that there are these patches? What is going on there? And so by looking at the distribution of EGFR in the cells, even without overexpression, you can see that there are these clonal effects in which EGFR is overexpressed all around it. And when we tried to titrate growth factors on top of these cells, what we discovered is that at physiological levels of the growth factors, only a fraction of the cells would respond. However, cells that are overexpressed, they all respond. So it is the low-level cell variability that can create these patches. And this is where I want to conclude by saying that I'm very, I hope that the type of techniques that are being developed and discussed here will allow us to understand that cell, why it is that these cells can pulse, can create these resistance cancer cells, not just with the staining of EGFR, but of all proteome at some point. So in conclusion, what I showed you is that we discovered some drug resistance mechanism that is due to receptor-driven pulsatile reactivation of the oncogenic pathway, which looks like this in time, looks like patches in cultures, and is due to the different pharmacology of oncogene and receptor-driven signaling. There are some questions, but of course needs to be followed up. What determines the single cell heterogeneity? How can we detect these in patients? And once we know what is the exact mechanism, can we add a combination treatment to curb this mechanism? I would like to thank Peter Sorger for the supervision, Chris especially for experiments creating the cell lines, Fabian Froelich for help with the modeling, for the proteomics at PNNL, Stephen Wiley and his group, and fundings.
Thank you, Luca. Marco has a question.
Hi, Luca. Great work. Short question. So about the patches. So is it always patches? Have you tried like this experiment where you try to keep them separate, treat them with the drug, and also you would get them up? Do they need this contact with each other to drive up EGFR receptor?
Yeah, no, we haven't tried that yet, and I think that would be pretty interesting to understand, because our hypothesis there is that there are some transient overexpressions, but is clonal because it happens to have a time of overexpression that is longer than the cell cycle. But that would be, I think, a great way to understand how much it is being in contact with each other or the ability to secrete these growth factors. So yeah, I think it's a great idea.
This is super interesting. I don't know if I missed it or if it's more like a curiosity question, but are those cells that still keep like the EGFR activity, or is there a correlation with their resistance to the therapy? So,
you mean in patients? Or
in patient or even in culture, like
So in cultures, we know that if we block this ability to do these pulses, then we have less cells re-entering the cell cycle. I haven't exactly done the nailing down experiments to follow these cells for as long as it takes to see the pulses and correlate to that, but I think that's the implication, that the pulses induce the cell division.
So on the slide where you showed the big pulse, it seemed that there was also synchronization between the cells. Is that I mean, do we know or do you have an idea what's, controlling this very tight coevent across this?
Yeah, so this is the question. What I've tackled here is mostly the ability of these cells to sense the growth factors, right? So, and the idea of the patch is that if they have the same high abundance of, let's say, EGFR, then they're going to be all respond to a signal. And the question now is, what is the signal? And when we look at this video, we can actually kind of triangulate, because in time you can actually see where it's originated from. And we are trying to study exactly what is the event, but we think there is a focal event that leads to the release of a signal, and that would explain why there is a synchronicity.
Hi, very nice talk. I have two questions. So the first question would be, have you checked the expression levels of ERK? Would it just because of the birthday of transcription, since it's also correlated with the cell cycle? So, and the second question is, I think you have a slide with both the ERK and MAC inhibitors together. So what do you see there? I'm just wondering. Thank you.
Yeah, so the first question was if, I guess, the cell variability in ERK could explain these differences. Did I get it right?
I mean the diversity of transcription. So instead of at the protein level, how about the mRNA level?
Yeah, so we definitely want to understand how much the variability we see is due to mRNA and therefore protein abundances. We haven't done anything in that direction at the moment, but we are definitely trying to maybe use RNA fish or this type of techniques to look at the RNA. And the other question was it, sorry? Couldn't keep it in mind.
The other question is about the ERK and MAC inhibitors, the double treatment.
So when we use MAC inhibitors to very high level, we can completely eliminate these pulses and then have around 20 times, 30 times less cells that re-enter the cell cycle. So there is something else there, not just the pulses of ERK, maybe some other signaling pathway that pulse together, because receptors could actually turn on not just ERK. But yes, the MEK inhibitors can deal with this type of reactivation.
Thank you, Luca.